Hi, I'm Chance,
And I'm Sarah Catherine,
and this is Conservation Connection.
presented by Last Chance Endeavors.
We run a wildlife education nonprofit focused on connecting students to their environment.
Each week here on Conservation Connection, we do just that by introducing you to the groundbreaking science and conservation work that's happening every day around the country.
We talk to professionals in the world of conservation science and wildlife management and ask them about their career,
Their current projects,
their wild and crazy stories from the field.
and everything in between.
Join us each week to discover just how these dedicated people are working to protect our planet. Today, we're talking with Dr. Mike Janech. He is one of the PIs of the COMPARE program, and he is an associate professor of biology here at College of Charleston.
Welcome to the show.
Thank you. Glad to be here.
We're really glad you're here too. So, I have to admit, when you sent us an email and told us what you did, Sarah Catherine and I had to sit down and look up a lot of words. One of the most important ones being proteomics, which is a big part of your day-to-day work, right?
That's correct.
So for people like me who had no idea what proteomics, or that proteomics is a field of study, can you give me a snapshot of what proteomics is?
Sure. So proteomics is the analysis of all the proteins you can possibly try to analyze in a sample at any one given time, including all the modifications on those proteins.
Okay, so basically, instead of genomics, where you're looking at the DNA, proteomics is looking at the proteins that are created based off of that DNA, right?
Exactly.
And it's different because DNA, genes in the DNA are expressed or not expressed. And so whether that gene is on or off is going to affect how much of that protein it's creating. So while the DNA can tell you what can be made, proteomics tells you how much was made of each different type of protein, right?
That's right. So the proteins actually execute the function of the genes, but it's really hard to predict from the gene itself, the sequence itself, how much of the protein is going to be there. And then further, how much of it's going to be modified that could adjust the activity of the protein. So either the protein could be made, but it's not doing anything, or it can be turned on by other molecules. And so that's what we really want to know with proteomics is not only how much is there, but how much of it could be turned on or modified, all these types of things.
Cool. So it's a much more holistic view of the mechanics of what's happening, opposed to the data behind it.
That's the attempt. We always attempt. So proteomics can never see everything, but we're attempting to see everything we can.
Or at least as much as possible.
As much as possible, yeah.
Okay, so that's really interesting. Very cool. So I don't think I even knew that this field existed. How did you get into this field?
So the field, way I got into it is I came back to South Carolina and got a job at the Medical University of South Carolina, and I was hired as the assistant director of the nephrology proteomics lab, and nephrology simply means study of kidney disease. And we were really interested in looking at the proteins in the urine from humans and classifying the different diseases based on the proteins that showed up in the urine. So you would go to a doctor, you could pee in a cup, run it through an analysis and say, oh, look, you have this particular protein is very high. You need this drug, or you need to see this doctor. And that's the way it all got started. And as I was going through this, I realized that proteomics and doing this big protein analyses like we would in humans wasn't really happening in animals, or it wasn't happening to the degree. And very few people were actually taking the data and saying, how does this align with a disease in an animal? What does this tell us about an animal? And if the environment is impacting health of an animal, could these proteins be really great indicators or biomarkers of disease or chronicity of disease?
So the idea is that if you have a specific disease, your body's going to react in a certain way, and the proteins in your cells and in different structures in your body will change in a predictable way. And so you can find, if you see a spike in this specific protein, you can link that to a specific disease that's causing that spike, right? So it's a diagnostic tool.
Right. It can be a diagnostic tool, or it can be a prognostic tool, which will tell you something about what's going to happen in the future to that particular patient or animal.
So you started doing this in humans, where obviously, because we are humans, there's a lot of people who are really interested in making sure that when you get sick, we can get you healthy again. But there's not as many, dolphin doctors as there are human doctors. So you were looking to make this technology sort of a more broadly available tool, right?
Correct. And that started with sea lions, actually.
Okay.
With the Marine Mammal Center in Sausalito, California, there's these big domoic acid outbreaks, which come from a diatom. And so that's just a small algae that lives in the ocean, and they'll produce this toxin that can get eaten by a little fish, and then the sea lions will eat those. And it causes seizures in the sea lions. It causes malnutrition. There's a lot of bad things that happen. They will actually die from this toxin.
Wow.
And so the problem is the toxin can enter the system, cause the damage, and then leave the system within 24 hours. So you don't know why this animal is shaking on the beach or seized, but it's seizing. So something must be wrong, but there's no toxin in the body at all. So we said maybe the information has been maintained in the proteins there, where something's happened to the body and they're making new proteins that then we can say, well, we can't see the toxin, but this protein wouldn't have been made unless the toxin was there and the body's trying to respond. And so therefore we could start following the proteins, the aftermath of it, to figure out what was happening earlier on.
It's a little bit like studying the wreckage of a tornado. The tornado's gone, but you can still track the path of it and kind of see where it was based off of how the buildings and the land has changed.
Exactly. And furthermore, you can take that information and say the body's responding this way. Are there any treatments? Is there something damaging that the body's doing to itself right now that we can stop? And so that's a lot of research and a lot of effort and takes a lot of time to get there. It's never overnight, and it's not as simple as it sounds. But the fact is that that's the whole intention of using proteomics. So we don't know which protein is going to go up or going to go down. I mean, it's really hard to predict this because the body is so complex. And, now you're talking the human body. Let's move it over to a sea lion. Well, what's going on in a sea lion? They're a little bit different. Dolphins are a little bit different. So we use proteomics to cast this really wide net, and we see where the signal is. And so we can ignore the noise and we go after the signals. And the signals are going to tell us something about the health of the animal, what the animal's doing. And is there any way that we can take advantage of that information to help the animal recover and then release the animal back out into the wild? So that's one of the ways that I got really interested with proteomics. And then from there, it just kind of developed. There's dolphins with metabolic syndrome and liver disease, but why is this happening? Do they have the same proteins that are going up as they do in humans with liver disease? We don't know. So let's study those and let's use proteomics to really encompass as much information as we can.
Yeah, absolutely.
So what are some of the things you're studying or researching currently?
So currently, there's a lot of projects going on in the lab. And because we do proteomics, it's not limited to one specific organism. Proteins are everywhere.
Yeah.
We tend to collaborate with a lot of investigators. So there are some investigators that are very fascinated to look at the interaction of bacteria and phytoplankton and look to see how they change each other's proteomes and what's going on there. And it'll tell us something about what's going to happen in the future. Are they going to stimulate growth? Are they terminating a bloom out in the ocean? So that's something we can do. I'm very interested in kidney disease in sea lions. That to me is fascinating because they do get a bacterial infection that does cause kidney disease. That's called leptospirosis. And humans can get the same thing, and humans do get the same thing. Dogs commonly get leptospirosis. But when you go out to a sea lion, how do you know it has the kidney disease? Do the markers that we use in the clinical lab, are they sensitive enough to show us that this animal has got kidney disease? And most of the time, the markers are pretty darn good, but they're not foolproof. And so what I like to do is to take my knowledge from the clinical side of research and apply it to this and the sea lions and see exactly what this is actually doing to the urine proteins, and if there's any proteins in there that are telling us, oh, look, it's got kidney disease, but we would not have seen it with traditional markers, and therefore it allows us to estimate the amount of kidney disease in a population better than using these traditional historical markers we relied upon for 50 years.
So it's really just working to make our knowledge and ability to diagnose issues in this population more robust, making our ability to find those issues and resolve them stronger, right?
It is. And that's one aspect. That's what I do. You can use proteomics to do other things. And I find it much easier. This is what I'm really interested in. And so whatever we find here, we can actually, using mass spectrometers, that's what the tools that we use to detect these proteins that we end up chopping up with peptidases, but that's not important. What's important is that we can take that information and we could rapidly create assays now where we couldn't before. And that's something that I think holds great benefit here. So instead of what I'm saying is, instead of saying that this protein is 2 times higher, compared to what, I don't know, we can go in there and say, this is how many micrograms per milliliter of sample there is of this protein. So we're basically able to standardize a measurement very quickly that can be used across labs, and they'll all know what the number is now, instead of saying 2-fold or 3-fold or 10-fold, which really it's a relative measurement that nobody knows what it means.
Nice.
So as you're researching this, I'd imagine there's 2 parts. So you're looking at, with proteomics, what is this telling us in this animal? Kind of like a diagnostic. Mm-hmm. And then are you also turning around and taking that and saying, okay, if there's this issue in this animal, what can we do to fix it or heal it or make it better? Is that part of it as well?
Yes. We try to do that, and that's called systems biology, where you take all of this information. So we're going to have maybe 10,000 data points here per sample. Okay. And then how do you make sense out of that? Well, there's computer programs now that know relationships between proteins. They know relationships between proteins and genes. And when you put the data into these computer programs, you start to develop a picture, and you say, oh, it's not just this gene and this protein that's involved, but there's a whole pathway of genes. There's a whole pathway of proteins. Well, what regulates that? And that's where you can start to think about, should we be interrupting that pathway? Because if we don't, then it's going to continue causing inflammation in the animal, and it's going to cause tissues to be destroyed and all this scarring to occur. And what we don't want is, we don't want an animal to continue scarring its organs, because that's ultimately going to lead to death.
Right.
So is there a way to interrupt that? So that's not a very simple thing to do. And that's what people do in human medicine all the time, is to take all of the data and say, exactly where should we be focusing, and can we manipulate that and mitigate the damage?
So you're taking this whole, 10,000 data points is a ridiculous number of data points in a single sample, and taking that information and trying to decide precisely where in this pathway could we introduce something that stops this signal from continuing, so that rather than it being this kind of runaway reaction that ultimately leads in the death of the organism, you stop it there, make one precision cut to stop that pathway, and the organism is able to recover much better.
Yeah, I think that's what everyone would like to do. That it's never that easy,
at least in theory, right?
Yeah, exactly. But if we have the data and we have the computers telling us where to go, at least we're not searching blindly anymore,
right?
And that's what's really critical with using these types of omics techniques. It could be genomics or transcriptomics, proteomics or metabolomics. All these omics techniques are trying to describe the system at any one time, and then what you like to do is be able to predict the system. And that's essentially what medicine in any field wants to do, is they want to be able to predict the system, that is, to predict what's going to happen to that particular patient. Are they going to respond to a therapy or not? And that's really critical, because you don't want to give somebody a toxic therapy when they're never going to respond to it. That's going to hurt the patient or the animal. So that's a very veterinary science focused approach. That's not kind of this big wildlife management approach. But you can certainly take the information, go to the mechanistic side, or you can go back and say, well, can we monitor over time what's happening to an animal in a way that it makes sense? That, is the liver getting worse over time in this population of animals versus that population that may be in a pristine environment? And then if we see this, we can say, well, why is the liver getting worse? Is there something in the environment that's causing the liver to get worse? But if you don't have those tests available, you're never going to ask that question. You're never going to see the damage until it's probably too late and the environment's really, really in distress and it's starting to affect humans.
Right. And especially because at least the model organisms we've been talking about, marine mammals like dolphins and sea lions, they're top predators for the most part. Maybe large sharks would be predating on them, but they're going to be some of the first organisms to show signs of an accumulated environmental toxin because they're at the top of the food chain. So they've got this bioaccumulation and biomagnification going on, where if there's heavy metals that get into the plankton, well, when that plankton gets eaten by fish, that gets eaten by a bigger fish, that gets eaten by a bigger fish, that ultimately gets eaten by a dolphin, that dolphin isn't eating one piece of plankton. It's eating hundreds of thousands of times that amount of plankton and the toxins that were in that plankton, because it's so much higher up in the food chain. And so having tools that can let us look at the organisms that are really susceptible to this kind of damage is really effective, and not just looking at the organisms themselves, but in using those organisms as sort of a canary in the coal mine of the environment that they live in.
That's correct. And importantly, there is the marine mammals are eating a lot of the same food that we're eating. It's the same fish species that they're preying on that we're actually preying on as well. So in addition, the marine mammals, the organ systems are extremely similar. Everything's very similar across mammals. It's far more similar than it is different.
Right.
And so what's critical there is that if we can see the damage in an animal that's in the environment and eating the same fish that we're going to eat, that's going to give us that whole canary in a coal mine, analogy there. It's going to give us some idea, what we could expect later on, and why is that happening, and perhaps we should focus in a certain area. Rather than, just kind of going across the whole United States, looking everywhere, we could say, hey, look, here's a signal. This animal's got some disease. We would have not picked it up because it's not dead yet. But can we mitigate this somehow, this particular area, whatever the insult is?
And how long have you been studying proteomics?
Proteomics for 13 years.
Okay. So I'd imagine a lot has happened in that time. What's kind of like the most exciting or game-changing kind of thing that you've seen happen?
Right. So not to get too analytical chemistry here, but I think that the fact that the mass spectrometers, the tools that we use, have gotten faster. They've gotten more accessible, and the technology has grown to where we no longer have the error in the measurements that we used to. We can have a lot more confidence in our results. And that, to me, has been probably the biggest game changer. And there's quite a few things that have also been done on the computational side to also address the errors in the measurements and the errors in the protein identification. I think those are two of the biggest without getting into too much detail.
Yeah, that's great.
So we've spent a lot of time talking about how proteomics can help with diagnostic and prognostic tools of marine mammals, but obviously, that's not the only application of this kind of science, and you're involved with a pretty big project that is making the data that comes out of this kind of work accessible to a wide variety of people asking a wide variety of questions. Can you tell us a little bit about the COMPARE program, including what COMPARE stands for?
Sure. Yeah. So COMPARE is called the Comparative Protein Aggregator Resource. And I know it's a big word, but we were kind of come up with something that was a really neat name. So it turned out to be COMPARE, but actually it's a really good description of what the project is. So what we want to do with COMPARE is compare all the proteomes of all the mammals. That would be the lifelong goal, and I'll be way dead before we can get to this goal. But what we'd like to do is start out with at least the mammals. And by comparing the proteomes, we're going to be able to compare what the differences are that you can't see. So not only, the genomes are really interesting to be compared, but the genomes don't tell us how much protein is going to be there. And you can look at the phenome, which is the phenotype of the animal, the thing you see, but it's pretty easy to count, how many horns something has or how many spines something has. But how much protein is in there, and does the amount of protein actually benefit a certain animal to do a certain physiological maneuver that we may not be able to do as humans? And the idea here is to start to connect all the animals in the world to humans and allow people to make these comparisons very easily amongst themselves and another animal that may be interested in. So as my co-principal investigator at the National Institute of Standards and Technology, who's also a partner on this particular COMPARE project, likes to say, everybody has their favorite animal when they're in kindergarten, and we tend to always have our favorite animal. And this is going to allow people to easily and quickly, compare themselves to that particular favorite animal. And we want to do this in a way. Cool. Yeah. So we totally want to do this in a way that it's available to everyone. It's accessible on a website, and it's not something that you have to become an analytical chemist to understand. It's more of a click button thing, and you'll be able to look at the differences in the proteins in the blood between yourself and it could be a tenric or a pangolin, or it could be a dolphin, and see how we differ. But what's most important with this is it also is going to allow professionals in the field who really understand something about dolphin physiology or vampire bat physiology to access this data without having to have millions of dollars of equipment and all this expertise. We'll do this for them, and they can quickly access the information. And we want to make it very simple for them to access.
And that will obviously make a huge difference. But before we move on, I feel like this is going to be ultimately, like, I feel like eventually we're going to get to a point and it's going to be like one of the DNA test kits where it's like, oh, let me send this in and I'm going to hear back like what animal like my DNA is close to. Or like, this is my favorite animal. Compare me to it. But yeah.
That would be fun. Yeah, I don't think we're going to do that.
Hey, maybe.
Well, feel free to steal my idea if you want to.
You need to pitch it to an investor to get some funding.
Definitely.
There you go.
Personalized proteomes. There you go.
So the idea here is kind of that if an organism is able to withstand an extreme event, so the loss of blood to a particular organ, but humans are not able to do that same thing, we can use this compare tool to see what proteins are maybe present in the organism that can withstand it and absent in the humans, and how can we use that to sort of look for clinical treatments that if it does happen to a human, we can use it to help them respond more effectively, right?
Right. So that's what you're getting at are things like ischemia reperfusion, what we would call in a diving marine mammal. So we have a marine mammal, and you can pick your favorite one. And when they dive, they actually shut blood flow off to certain organs. So things like that don't typically happen in humans unless you're on an operating table, and it shouldn't happen, but it does. And if something happens where blood flow is diverted from an organ for a significant amount of time, the organ will become injured. Sometimes the organ recovers, and sometimes the organ doesn't recover. And if it doesn't recover, that becomes a chronic disorder that has to be treated over, many, many years. And that really is a detriment to the patient. So what can we learn from an animal that actually can divert blood flow from an organ and reestablish blood flow to an organ but doesn't get injured? And can we learn something from that particular animal and apply it to a scenario where we have a patient in an operating room where blood flow is diverted? Or you could think of this in a way like a heart attack would be one of the ways where blood flow actually no longer goes into the heart because it's clogged, and therefore you get the damage in the heart, and then you have to reestablish the blood flow. But when you do, the heart's never the same. Right. So, diving marine mammal can do this, and they're perfectly happy.
So maybe we can learn a trick from them.
Yeah, and so our idea is that, what are the differences? What are the basic differences? And when I started the project, it was quite amazing. I just was thinking, well, what are the major proteins in the blood of a dolphin? And we don't know. We have no idea. And you say, okay, well, what are the major proteins in the blood of a pig? What are the major proteins in the blood of a cow? And the more and more you ask these questions, you look, there's no data out there. And so we started to say, let's start filling in these gaps and just start to know what makes a cow, and what makes a pig, and what makes a dolphin, and a human. And as we start to look at these, what I've really appreciated was it's not so much how we're different, it's how similar we are. We are extremely similar to all these other animals. But there are some small differences, and these small differences are fascinating differences.
And in turn, I guess, make a big difference in some ways.
That would be the hope. Yeah. So.
What's been the biggest surprise so far in your work with the Compare project?
So it was, we had a graduate student on this, Bailey Boxa, who just graduated from the Grice Marine Lab program. And she was focused on a protein that we had published a few years back that was really, really high in dolphins, but not very high in humans or rats or pigs. And this protein has gotten quite a bit of bad press because it's a protein called vannin that actually converts panthenine to pantothenic acids or vitamin B5. So, it's an important protein. It's pretty common, but really high levels of this protein were associated with inflammation and all these bad things, liver fibrosis. This is really bad. And here we are, we have this protein called vannin staring us in the face that's really, really high in dolphins. It's like at least 100 or sometimes 1,000 times higher than it is in humans.
Wow.
And so you're going, wait a minute, what's going on here? So this protein also has a pretty good thing. It actually makes free thiol, which is just a sulfur group. And these free thiols are really reactive and they can act as antioxidants. And so we thought, well, perhaps there's something to this. Maybe there's a lot of antioxidant coming from this protein being able to slice up these metabolites in a certain way. And so we thought it's only going to be in diving marine mammals. And as we profiled, or she actually profiled out 57 different mammals across a phylogenetic tree, we found that no, vannin actually is something that's not unique to just divers or something that gives them, the ability to dive. No, vannin is actually huge across anything with a hoof. So that's what was surprising to me. I was not expecting that. I don't think anyone was expecting that. And we're going to write those results up and publish them soon. But to me, it was just fascinating. Here we've got this protein that if we just look at carnivores, it's not very high, and therefore it's probably associated with disease if it's high. And then you look across the whole, phylogenetic tree of mammals, and no, it's not just a low protein. It is really high and defines a certain group of mammals that we would have never expected before. And so I think that's really, really pretty cool.
Yeah.
That's absolutely fascinating. So correct me if I'm wrong, but dolphins don't have hooves.
No.
Okay,
No,
but
so. They're
part of
descended.
the group Artiodactylate, right?
That's correct.
Did I get that word right?
That's right.
Okay.
Yeah.
So that means that dolphins and whales, as we know them today, before they became what we see swimming out in the ocean, their ancestors are also the ancestors of the hoofed animals that we see on Earth today, right?
That's correct.
That's pretty crazy that you can see that evolutionary history in something as highly evolved as a dolphin, and it's still shared by its hoofed relatives.
Yeah, that's what is really neat. So the idea that this high vannin 1 phenotype, as we were calling it, would have evolved in the marine mammals isn't true at all. It would have started back a long, long time, greater than 60 million years ago, according to this phylogenetic tree. So why? And that's where we're at this particular point. Why the heck is it so high? But it's something we would have never even thought about had we not just looked at the proteomes across these particular mammals. And that's not the only one. So there's a number of other proteins that have different distributions. Our problem now is we don't have enough mammals. We want as many mammals as there are, and there's something pretty close to 5,000 different species of mammals. So I think there's a lot of stories here that are floating around in the blood of these mammals, and we can use proteomics to get at those stories and better understand the relationships between the mammals, and then maybe some of the pathways and physiologies that are related to these particular proteins that are floating around in the blood.
That's amazing. You're answering questions that you didn't even know you had.
That's the whole idea behind it, is to show us what we don't know we don't know.
Yeah.
That's exactly it's kind of weird to say, I know.
That's absolutely fascinating. Now, for maybe some of our listeners or anyone who wants to get involved in this field, because, as we've been sitting here talking with you, it really is fascinating and so broad, and there's so much that can be done with it. What's something you might tell them if they're looking to get into this field or do work related to proteomics?
So I would say number one for any student that's interested is be curious and then be passionate. Those are the two biggest things. If you're curious, people are going to respond. Teachers are going to respond. Professors are going to respond to you. So those, I would say, now, if you're specific with proteomics, there are proteomic labs. So when you enroll in a school somewhere and you enroll in a school that has a proteomics program or proteomics core facility, establish a relationship with the proteomics investigators that are there. And I'm sure they will welcome students that are interested in proteomics. There's not a whole lot of students interested, clamoring to get into proteomics, but we're always looking for really good students. A chemistry background is always very, very helpful. Math is very helpful, but you don't have to be a chemist or a mathematician. You can be very interested in computer science and get into proteomics, because most of the work in proteomics is on the computer. And I can tell you right now that I wish we had more computer scientists interested in proteomics or any omics, because that's where the discoveries are going to be made. It's very difficult to make these connections, and there's a lot of connections. There's a lot of network, a lot of systems biology, a lot of data analysis. And so you don't have to be this biologist going straight, to proteomics. You can come into it from many different angles.
Awesome. Well, thank you so much for taking the time to sit down and talk with us. I have learned a lot today, and interviewing you. It's opened up a whole new world that I didn't really know existed.
Yeah, absolutely. Thank you for being on the show today.
Thanks for having me. This was fun.
Thanks for listening to this episode of Conservation Connection.
If you enjoyed our podcast, go ahead and subscribe to make sure you catch every episode that we post.
We'd love to hear from you. So if you want to reach out, go to our website, lastchanceendeavors.com/contact, and shoot us an email.
We love questions from our listeners, so if you heard something that you want to know more about, be sure to let us know.
If you've got a minute to spare, leaving a 5-star review on Apple Podcasts will help other conservation-minded people find the show. We'd really appreciate it.
A big thanks to the people working to protect our planet, and a big thanks to you for listening. Don't forget to tune in next week.